if you open up almost any general biology textbook and just like flip to the chapter on the cell, you get this very specific, almost comforting picture.
Oh, yeah, the classic color-coded diagrams.
Right, where
Yep.
everything is neatly in its place. And right there in the middle, usually colored, vibrant purple or something, is the nucleus.
Yeah, always dead center.
And the textbook almost always labels it with the exact same phrase, which is the cell's control center, like the CEO's office.
Which gives you this visual of a mahogany desk where the nucleus sits, right? Just issuing these top-down orders to the rest of the cell.
Exactly. But as we launch into our deep dive today into the nucleus and our DNA, and welcome, by the way, to you, the listener, as we unpack this, I have to point out that our sources suggest this whole CEO metaphor is, well, severely outdated.
It really is. I mean, it's a tidy, functional way to teach it, sure. You establish it as the brain of the operation so people grasp its importance.
Right. But the modern biological picture seems far messier, highly reactive, and honestly much more fascinating. So I want to start by challenging you on this right out of the gate. Why is that whole control center idea considered a failure today?
Well, it fails because it implies a one-way street of command. When we actually observe how a cell operates, the nucleus isn't sitting behind a closed door making unilateral decisions.
It's not just barking orders.
Not at all. It functions much more like a massive hub of biochemical negotiation. It rarely just issues commands. Usually it reacts.
Reacts to what?
To a constant, overwhelming influx of signals arriving from outside the nucleus, specifically from the cytoplasm. We're talking about, hormones, growth factors, metabolic stress signals.
Oh, wow.
Yeah, and those external signals cross into the nucleus and actually dictate which genes are read and which stay completely silent.
Okay, let me see if I have this straight. It's less like a CEO's rigid blueprint that must be followed and more like a massive restaurant recipe book.
I like that.
And the kitchen's current environment, like whether they're in the middle of a chaotic dinner rush or just prepping for a quiet morning, is what dictates which recipe actually gets pulled off the shelf and cooked.
That analogy works perfectly. But we actually need to take it a step further to appreciate the sheer complexity here, because it's not just a matter of reading a recipe cover to cover.
Right, and this is where I actually got a bit lost in the research. Because when I learned this stuff, there was always that phrase, one gene, one protein, you read the recipe, you get the exact dish.
The classic central dogma, yeah.
But the sources keep mentioning things like alternative splicing and epigenetics. And I know they complicate the picture, but I need you to explain how they actually work mechanically. Like, what is alternative splicing?
Let's tackle that first. So imagine your recipe for a soup has 10 paragraphs. In a simple, one gene, one protein world, you read all 10 paragraphs and you make the soup.
Right.
But human cells have machinery that can edit the instructions after they're copied from the DNA. This editing process can physically cut out, say, paragraph 3 and paragraph 7, and then splice the rest together.
Wait, really? Just skip parts entirely?
Exactly. Splicing them together to make a slightly different set of instructions. So one single gene might yield 3 or 4 different variations of a protein, all depending on how those instructions are spliced together.
So one gene is not just one protein, it's more like a flexible template.
Exactly.
Okay, what about epigenetics? Because that word gets thrown around a lot. How does that physically change things?
So epigenetics involves actual chemical tags that attach directly to the DNA or to the proteins that the DNA is wrapped around. And these tags, they don't change the genetic sequence itself.
They don't rewrite the code.
Right. Instead, they change how tightly the DNA is spooled.
Yeah.
If a section of DNA is spooled too tightly because of a specific chemical tag, the cell's machinery physically cannot get in there to read it.
It's blocked.
Yep, that gene is turned off. But if the tag loosens the spooling, the gene opens up and becomes readable. It's essentially an environmental response mechanism.
That makes so much more sense. It's physically hiding or revealing the text. And the real-world proof of this is wild. I mean, the sources mention identical twins.
Right, classic example.
They start out with essentially the exact same DNA sequence, right?
Yes, the foundational code is practically identical.
But over time, they can develop entirely different diseases, exhibit different personalities, and their fingerprints aren't even perfectly identical. And that is all because their different life experiences, diet, stress, environment, are applying different chemical tags.
Exactly. Their epigenetics are different. And, you don't even have to look at twins to see this. Just look inside your own body.
What do you mean?
Well, a liver cell and a neuron in your brain contain the exact same genetic library, the same genome.
Oh, right. I always forget that.
Yeah, but a neuron looks like a sprawling tree and transmits electricity, while a litter cell is basically a biological filtration plant. They look and act completely differently because their epigenetic tags have permanently locked away different chapters of that library.
So the neuron is literally just blocked from reading the liver cell instructions.
Precisely, forcing them to only read the instructions relevant to their specific jobs.
Okay, so if the nucleus is this vast reactive library holding the recipes for an entire human being,
Mm.
we have to talk about the physical space, because the math of the compaction here just breaks my brain.
It is pretty staggering.
How does a cell physically store that much information in such a microscopic space?
Well, it requires a level of physical compaction that is almost unparalleled in nature. Let's look at the baseline numbers. In a single human somatic cell, which is just a standard body cell, there are roughly 3.2 billion base pairs of DNA.
3.2 billion in one cell.
Yes. Organized into 46 chromosomes, or 23 pairs. And if you were to somehow extract the DNA from just one of your microscopic cells and stretch it out end to end, it would measure about 2 meters long.
2 meters. I mean, that is taller than I am. Inside a single invisible cell.
And that 2 meters of material has to be housed inside a nucleus that is typically only 5 to 10 micrometers across. We are talking about a compaction ratio on the order of 200,000 to 1.
See, if I take 2 meters of string and just shove it into a microscopic ball, I'm going to get a permanent, unusable knot.
You'd never untangle it.
Right. If this is a library that needs to be constantly read and referenced, how does it not just tangle into chaos?
Well, it avoids tangling because it isn't just a loose string. It relies on those spools we mentioned earlier. The DNA double helix is meticulously wound around specialized 8-part protein complexes called histones.
Okay.
This is often described visually as beads on a string, which is a structure we call a nucleosome.
So the histones act like tiny sewing spools, keeping the thread organized.
Exactly. And those beaded strings coil themselves into thicker fibers, creating a dense but organized material called chromatin. And this chromatin is the everyday working state of your DNA.
Not the little X shapes.
No, actually. The only time it ever condenses down into those dense X-shaped chromosomes you see in textbooks is when the cell is actually dividing and needs to move the DNA around safely without breaking it.
It's incredibly elegant. But a library this precious, carrying all that compacted information, clearly needs heavy protection. Which brings us to the walls of the nucleus, the nuclear envelope.
Right.
But reading through the research, it seems like treating it as a simple brick wall is another textbook oversimplification.
Oh, it is much more dynamic than a wall.
Yeah.
The nucleus is enclosed by a double membrane structure, the nuclear envelope, but it is heavily fortified and highly interactive. It's studded with thousands of structures called nuclear pore complexes.
And these are the gates.
Right, but they are not just passive holes in a wall. They are massive, highly intricate protein machines.
When you say massive, we're talking on a cellular scale.
Right. Yes, but even on an atomic scale, they are absolute behemoths. Biologists measure molecular mass in daltons, which is roughly the mass of a single hydrogen atom. A typical protein might be a few thousand daltons.
Okay.
A single nuclear pore complex has a mass in the tens of millions of daltons. It is constructed from about 30 distinct proteins, called nucleoporins, arranged in this massive symmetrical ring.
That sounds like a serious heavy-duty security gate.
Yeah.
So how selective are these gates? Because we established earlier there's a constant flow of signals going in and out.
The traffic control is intense. Small molecules like water or basic ions, they can pass through the pore freely, but the pore complex acts like a bouncer when it comes to larger cargo.
A bouncer?
Yeah. If a protein is larger than about 40 kilodaltons, it hits a physical barrier, a mesh-like tangle of proteins inside the pore. It cannot just wander through. It requires a specific VIP pass.
How does a molecule get a VIP pass in a cell?
It needs a specific sequence of amino acids built into it, called a nuclear localization signal. There are specialized carrier proteins floating in the cell that recognize this signal, bind to the molecule, and interact with the mesh inside the pore.
Oh, wow.
They essentially negotiate with the pore, causing it to physically shift shape and shuttle the larger molecule through.
That is fascinating. But here is what really threw me during my prep for this deep dive. We are talking about these double membranes and strict gatekeepers. And usually we think functional compartments in biology require a physical boundary.
Right.
But the nucleus actually contains functional rooms without walls.
Ah, yes.
Yeah.
You are referring to the nucleolus, and you're right. Understanding how it works forced cell biologists to rethink some fundamental assumptions in the 2010s.
The nucleolus is that darker, really dense spot inside the nucleus you see on textbook diagrams, right? What exactly is it doing in there?
It is essentially a factory subregion. Its main job is to transcribe and assemble ribosomal RNA. It builds the subunits for ribosomes, which are the machines the cell will eventually use to manufacture proteins.
Okay.
But the twist, as you noted, is that the nucleolus has no membrane of its own. There is no physical wall separating this factory from the rest of the genetic library.
Which raises the obvious question, if there's no wall holding it in, why doesn't the nucleolus just dissolve and diffuse into the surrounding fluid?
It stays together purely through physical chemistry via a phenomenon called liquid phase separation.
Okay, you're going to have to explain that to me. Is that like shaking up a bottle of oil and vinegar dressing?
That is actually a very accurate way to visualize it. Just as oil molecules prefer to interact with each other rather than the surrounding water, certain proteins and RNA molecules inside the nucleus have a chemical preference for sticking to one another.
Oh, I see.
Yeah, when their concentration gets high enough, they spontaneously condense into a droplet. It is a dynamic functional compartment that self-assembles without a physical barrier.
Membraneless magic. I love it. But let's step back out to that physical wall we discussed earlier, the nuclear envelope. Because that physical separation defines one of the biggest rules in biology, the central dogma.
The flow of genetic information, DNA to RNA to protein.
Right, and there is a huge misconception that the nucleus itself makes proteins. But the nuclear envelope physically prevents that, doesn't it?
It does. It separates the process into two distinct geographic zones. Inside the nucleus, the DNA code is transcribed into messenger RNA, or mRNA. That mRNA gets heavily processed. This is where the alternative splicing we talked about earlier happens.
Right, the cutting and pasting.
Exactly. Then, that finished mRNA instruction manual is exported through those heavy-duty nuclear pores.
Out into the cytoplasm.
Correct. It is only out in the cytoplasm that the ribosomes read the mRNA and translate it into a physical protein. The nucleus produces the blueprints. The actual manufacturing happens on the factory floor outside.
And just to show how vital that wall is, we can look at bacteria.
Right.
Because they don't have a nucleus.
Right. Bacteria are prokaryotes. They have no nuclear envelope. Their DNA sits in an open region called a nucleoid.
So it's all just mixed together.
Pretty much. Because there's no wall, transcription and translation happen in the exact same space. A ribosome might literally attach to an mRNA strand and start building a protein while that mRNA is still being copied from the DNA. It all happens simultaneously.
It sounds so chaotic. Our cells seem so beautifully orchestrated in comparison. Okay, so we've established this incredibly protected, compacted library that selectively sends out messages based on environmental cues. But I want to talk about how we actually proved that this highly specialized system works the way we think it does.
Right, the experimental evidence.
The source has brought up Dolly the sheep, and the sheer persistence of that experiment really stuck with me.
Well, Dolly is the ultimate proof of concept for the genetic library. This was 1996 at the Roslin Institute in Scotland. Researchers Ian Wilmot and Keith Campbell performed a seemingly impossible biological feat. They took a nucleus from a mammary gland cell of a 6-year-old adult sheep.
Meaning, a cell that had already fully specialized, its epigenetic tags were fully set to be a mammary cell.
Exactly. They extracted that mature nucleus and inserted it into an empty egg cell. And after 277 failed attempts, they finally achieved one live birth. Dolly.
Wait, 277 attempts?
277, yeah.
When I read that number, it just highlighted how incredibly difficult it is to reset those epigenetic tags. But Dolly's existence fundamentally proves something about the blueprint, doesn't it?
It proved that specialization is not destruction. Even though that mammary cell had spent years only reading the mammary gland chapter of the DNA, it had not discarded or permanently destroyed the rest of the genome.
It was all still there.
Exactly. The entire intact code needed to build a brand new complex organism was still sitting there inside the nucleus, just waiting for the right cellular environment to read it again.
It is amazing, and it seems so obvious to us now, right? The nucleus holds the ultimate intact code for life. But digging into the history in these sources, getting to this realization was a messy, centuries-long scavenger hunt. People were just stumbling around in the dark for a long time.
They were looking at the answers without knowing the questions. In the 1680s, the Dutch microscopist Antony van Leeuwenhoek observed nucleus-like structures in the red blood cells of salmon, but he had absolutely no concept of their function.
Just a blob under a microscope.
Right. It wasn't until 1831 that a Scottish botanist named Robert Brown, who was observing orchid cells, repeatedly noticed an opaque spot. He coined the term nucleus from the Latin word for little nut or kernel.
But again, to him, it was just a visual feature. He didn't know what it did.
Correct. The actual chemical breakthrough came a few decades later in 1869, and the methodology was far from glamorous.
I am so glad you're bringing this up because this is my absolute favorite, weirdest piece of science history.
So a Swiss physician named Friedrich Miescher was working in Germany trying to isolate the chemical components of cells. He decided to study white blood cells because they have large nuclei. To get a steady supply of white blood cells, he went to a local surgical clinic and collected their discarded, pus-soaked surgical bandages.
I can't imagine being the person washing pus out of bandages in an 1860s laboratory, but it worked.
It did. Pus is incredibly rich in white blood cells. Miescher managed to isolate a unique phosphorus-rich substance from those nuclei that resisted protein-digesting enzymes. He called it nuclein.
Which was DNA. He literally held DNA.
He discovered DNA, but the profound historical irony is that Miescher himself didn't believe nuclein carried heredity. At the time, the scientific consensus was overwhelmingly in favor of proteins being the genetic material.
Why are they so convinced it was proteins?
It was a matter of perceived complexity. DNA only has a 4-letter chemical alphabet: adenine, thymine, guanine, and cytosine. Proteins, however, are built from 20 different amino acids.
Ah, so it's a math problem.
Right. Scientists reason that the vast, staggering complexity of life could only be encoded by a highly complex 20-letter alphabet, not a simple 4-letter one. DNA was dismissed as mere structural scaffolding.
That makes logical sense for the time, actually. So how did the scientific community finally get convinced otherwise?
It took some really ingenious experiments in the mid-20th century. First, the Avery McLeod-McCarty experiment in 1944 demonstrated that purified DNA alone could transform harmless bacteria into deadly ones.
Wow.
But the final nail in the coffin was the Hershey-Chase experiment in 1952. They used bacteriophages, which are viruses that infect bacteria. By using radioactive tags, they proved that when a virus attacks a cell, it leaves its complex protein coat outside.
So the protein doesn't even go in.
Exactly. Only the virus's DNA is injected into the bacteria to hijack the cell and build new viruses.
That is brilliant. The protein never even enters the cell, so it can't be the blueprint. And that, of course, sets the stage for the grand finale of the structural mystery, which is 1953, when James Watson and Francis Crick published their paper resolving the double helix structure.
But we absolutely must contextualize that triumph. Their theoretical model depended critically on experimental X-ray diffraction data that they did not generate themselves.
You're talking about Photo 51.
Yes. Photo 51 was an astonishingly clear X-ray image of DNA produced by Rosalind Franklin and her student Raymond Gosling at King's College London. That image was shown to Watson by a colleague, Maurice Wilkins, without Franklin's direct knowledge.
Wow.
Watson, Crick, and Wilkins went on to win the Nobel Prize in 1962. Franklin had tragically died of ovarian cancer in 1958, and the Nobel is not awarded posthumously. Today, historians rightly view her as an essential, deeply under-acknowledged architect of this discovery.
It's an incredible story of discovery, but I think we also have to acknowledge that treating DNA as an ultimate unchangeable code led to some very dark historical moments. Because if society views the nucleus as a rigid, deterministic destiny, things can go horribly wrong.
It is a sobering part of the history we must address. In the early 20th century, the emergent understanding of genetics was violently distorted into the pseudoscientific movement of eugenics.
Right.
The flawed idea was that human traits and societal value were strictly determined by a fixed genetic essence.
And looking at the facts from the source material, this wasn't just fringe theory. It resulted in horrific state-sponsored actions. In the United States alone, over 60,000 people were subjected to forced sterilization programs between the 1900s and the 1970s. And of course, these concepts formed the ideological foundation of Nazi Germany's racial hygiene programs.
It highlights the extreme danger of biological determinism. The tragic irony is that modern genomic science completely refutes the foundation of those movements. We know definitively today that genetic variation within any given human population vastly exceeds the average genetic differences between different populations.
So the science just flat out disproves it.
The science simply does not support the bigotry.
It's a dark reality we have to keep in mind. And it's interesting because The desire to view DNA as a perfect, clean, highly efficient blueprint that was shattered again by the genome itself. I'm thinking of the 1970s, when scientists realized that only a tiny fraction of our DNA actually codes for proteins,
Right. Only about 1 to 2% of the human genome consists of protein-coding genes.
which led to the rest of it, the other 98% being famously labeled junk DNA, like evolutionary debris. But is that still the consensus today? Is 98% of my DNA really doing absolutely nothing?
This is one of the most fiercely contested frontiers in genomics right now. In 2012, a massive international research effort called the ENCODE project published data suggesting that a massive portion of that 98% actually exhibits some form of biochemical activity.
Like what?
For example, they found that RNA polymerase was transcribing it, or that regulatory proteins were physically binding to it.
Oh. So it isn't junk, it's actively doing things.
Well, this is exactly where the debate ignited. Critics immediately pushed back on the ENCODE findings. Their argument relies on the mechanism of evolution. Just because a piece of DNA has a protein temporarily bump into it and bind, or just because it gets transcribed, does not mean that activity is vital for the organism's survival.
So it could just be an accident.
They argue that much of this activity could just be biological noise, random biochemical reactions with no functional importance. Resolving how much of the genome is truly functional versus merely active noise is a very live debate today.
That really drives home how much we still don't know. And speaking of things that defy our basic assumptions, we've spent this entire deep dive establishing how crucial the nucleus is. Does literally every cell in the human body have one?
No, there are fascinating exceptions. The most prominent example is your mature red blood cells. As they develop in your bone marrow, they undergo a process to physically expel their nucleus entirely.
They just eject their own reference library. Why would a cell do that?
It's an extreme evolutionary trade-off for efficiency. By ejecting the bulky nucleus, the cell frees up maximum internal volume to pack in hemoglobin, the protein that carries oxygen.
Ah, so it's all about making room.
Right, but the downside is massive. Without a nucleus, the cell has no DNA. It cannot divide, it cannot manufacture new proteins to repair itself, and as a result, it only survives for about 120 days circulating in your bloodstream before it breaks down and has to be replaced.
So this little nut, as Robert Brown called it really is the literal foundation of so much of what we do in modern science and medicine. I mean, think about the real-world applications for you listening to this right now. If you've ever spat in a tube for an at-home ancestry kit, the lab is actively extracting and sequencing the DNA from the nuclei of the cheek cells in your saliva.
Absolutely. The applications are everywhere. In medicine, directly reading the sequence of nuclear DNA is how we test for severe disease risks. Testing for mutations in the BRCA1 or BRCA2 genes to assess breast and ovarian cancer risk relies entirely on this science.
Because it's written right there in the code.
Exactly. Cancer itself is fundamentally a disease of nuclear DNA damage mutations that disable the cell's normal checks on division, leading to uncontrolled growth.
And it stretches far beyond the hospital, too. Forensic DNA fingerprinting, which was developed by Alec Jeffreys back in 1984, compares specific variable sequences in the nucleus to identify individuals. That exact science is the cornerstone of what the Innocence Project uses to exonerate the wrongfully convicted today.
It's revolutionized justice.
And looking to the future, we have the 2020 Nobel Prize, awarded to Jennifer Doudna and Emmanuelle Charpentier for CRISPR-Cas9. That is literally a technology that works by acting as molecular scissors, cutting and modifying the DNA directly inside the living nucleus.
It is a stunning trajectory. We have moved from barely being able to see this tiny structure under a microscope to deducing its code to now actively possessing the tools to edit the reference library itself.
It really is staggering. Well, as we wrap up this deep dive, I want to thank you, the listener, for coming along with us as we journeyed from those early microscopes to pus-soaked bandages to the absolute frontier of gene editing. But I want to leave you with a final thought. Something to mull over on your own that builds on what we discussed today about the power of epigenetics.
It's the perfect angle to conclude on.
If the sequence of your DNA is exactly the same in almost every cell in your body, and it's essentially identical to a twin if you had one, yet it yields completely different behaviors, diseases, and risks based on the environmental signals surrounding it, how much of your destiny is really written in the rigid ink of your DNA, and how much is ultimately decided by the way your life chooses to read it?